So we have the pleasure to have with us today Filippo del Bene. He studied industrial biotechnology at the University of Bologna. He did afterwards a PhD at EMBL with Johan Whitroot. Then he moved to the States to the lab of Erwin Bayer at the University of California, San Francisco. And afterwards, in 2010, he started his independent lab at the Institut in Paris. And in 2019, he moved to the Institut de la Vision. So, Filippo, thank you so much for your time to answer our questions. Can you tell us, in summary, your research interest?
So I'm one of the scientists that I have a very broad research interests. If you want to focus on one, is the development and function of the visual system. And I studied that using zebrafish mostly as a model system. But from there, we also go to the technology development with CRISPR-Cas9 tools development, evolution, comparative approaches, and so on.
Very interesting. And how did you realize that you wanted to be a scientist?
So actually this one is quite funny being in Spain saying that. I grew up and lived for the first 24 years of my life in a street in Italy, small, my hometown, called Golgi Street.
No
which,
way.
Yes, which, here with Cahal Golgi thing. And then as a child I was asking, Mom, who is Golgi? Why they give a name of streets? Like, I was a famous scientist. And then back then I already had a big passion for animals and studying plants and studying nature. And then I had my first aquarium when I was 10 years old. So I always knew I wanted to study animals and biology. So that's it. And, Golgi was kind of my mind. I think now I like Kahal more, but that's it.
So interesting. And how did you develop an interest in the study of the visual system?
So when I started my PhD, I had this amazing opportunity to work in the lab of Jochen at the MBL in Heidelberg. And his main interest was the visual system already. And for me, I was so excited to be able to work with fish. And then I got hooked up in neuroscience because, the visual system for me is a model to understand the sensory integration, sensory perception, how the brain works, how the brain interprets the environment around itself, and how the brain is formed. So, and then I kind of came natural to stick with that all my life. And now I'm really passionate. And among the senses, I think vision is the one that we can relate more because we depend so much on it. It's really the key sense for humans.
Exactly, another animals. And during your adult life, you moved across countries, right? So you did your PhD in Germany, then you did a postdoc in the US, and now you work in France. How this influenced you as a researcher and as a person?
Well, as a researcher, I think I try to be open-minded and understand that each country, each system has the positive and negative sides. So I try not to complain too much always, where colleagues always complain. I always tell them, look, it's not so rosy on the other side. There are problems even there. You just don't know, but this keeps me in a positive attitude in appreciating the different systems. And as a person, gave me the opportunity to meet a lot of great friends, a lot of fantastic colleagues all over the world, but at the same time it's a bit hard because having moved in my 20s and 30s every 5 years quite regularly, then you don't live in the same city where these great friends are. You can travel, you can see them once a year, once every 2 years, but it's a bit hard not to have them close.
I understand you. So during your career, you obtained several funding, and in particular, several ERC grants that are very competitive. How this funding impacted your lab and your research?
Well, the ERC, because it's it's on a 5-6 year time scale, and it's a lot of money for the European system, gives you first of all two things. On a practical level, it gives you the opportunity to hire people, to develop ideas, and even if the ERC is given for a specific project, you can a little bit expand on that project and explore avenues that you wouldn't have done otherwise. And that's also because having the security of funding for quite a long time, it puts you in in a state of tranquility and a little bit more relaxed, where you can be try to be more creative without the urgency of, getting the next grant for next year and so on and so forth. So it's definitely a great thing. It's a luxury to have. And the good thing of the ERC is really that they want to target groundbreaking, but still basic research. So it's not, it's really blue sky research, high risk, high gain, and it's the best part of my job for me.
Very interesting. So the last question, what would be the question that you would like to answer during your career?
Well, I think I will stay on the visual system mostly, and I will stay using fish, zebrafish, and other models. So I would like to understand how vision is evolved in the animal kingdom and how it's adapted to the different conditions, and how the interplay between developmental biology and neuroscience is regulated to adapt to this different situation where the visual system has to perform. And if in the meantime, if I can always promote fish as a model system in the scientific community, I will be very happy.
Exactly. Very cool. Very interesting. Thank you so much, Filippo.
Thanks to you.